Good afternoon, students. My name is Harender, and I am a PMR fellow from Department of Biological Sciences and Bioengineering from IIT Kanpur. So we have been trying to understand the basic principles and processes that have been involved in biotechnology. So this is chapter 9 of class 12 biology. So till now we have been discussing about the tools of recombinant DNA technology, which is one of the major part of biotechnology itself. And in the tools of recombinant biotechnology, we have already covered the concepts of restriction enzymes, of what they are, how they are, and from where they're extracted, and what they are used as. And then we have also learned about cloning vectors, the features of cloning vectors, why, how they're very important, and how they're basically able to transfer the gene of interest from like in our desired organism to get our desired product. So we have learned thoroughly about cloning vectors. Now in this session, we'll be discussing something we'll be discussing about the competent host, and then after that, after the discussion of this competent host, we'll be basically summarizing everything that we have learned right now, and then we'll answer a few questions so that the understanding of the concept is like clearer to you. So let's continue. Already discussed. We have discussed what? We have discussed about molecular scissors. Scissors. So this is the first tool where from this we are getting our desired gene of interest because we are cutting a gene of interest, and then we used our cloning vectors. Cloning vectors. So these are basically examples. The most common example that we learned was plasmid. So the gene of interest and the cloning vector. Then this molecular scissor will cut both of them, and then using the ligase enzyme, we'll basically stick them together. So we'll have a complete plasmid with a gene of interest. The gene of now this gene of interest needs to be amplified, or even like maybe like let's say we know that from the DNA we get RNA, from RNA we get a protein, right? Central dogma of life. This is central dogma of life. So this is known, this is well known, this has been known for more than 70, 80 years now, 56 years now. Yeah. So now we know this now. Now we have introduced the gene of interest. But now what is the purpose? Why are we introducing the gene of interest into this host? Sorry, into this vector? Like I mean, what is our end goal? What is our end product? Now we know that from every DNA, this is what we are doing. We are putting the gene. From this, we are getting the final product is protein. So based on that, we'll understand what is our desire. Like what do we desire? Like why are we cloning this? So this vector cannot say this is just one. This after ligation, you are just getting only one vector. But you want to mass produce this gene, maybe like for example, let's say, let me give you a practical example that you understand that diabetes is one of the leading causes of death worldwide. Diabetes is the leading cause of death worldwide, and in this diabetes, there is like one of the most common diabetes is type 1 diabetes. Type 1 diabetes, wherein when there is no insulin production, right? Insulin production in the body. So because it has affected the pancreas. So like we won't go in that deep. So we understand that diabetes is one of the leading causes of death. And in the type like diabetes, there are like multiple types of diabetes, but we are here discussing about type 1 diabetes. So you might have heard people or any relative or anyone's uncle, anything, that they take insulin shots, insulin injection or shots, injection, right? Now insulin again is a protein, it's a protein, right? So what people are like mostly the pioneering of was Eli Lilly. Eli Lilly company was the one who was responsible in synthesizing this insulin from really like from the bacteria and then basically commercializing it. Before Eli Lilly, before Eli Lilly, basically we used to get this insulin from pig's gut, from pigs basically. So this was the main source of insulin. Before that, we used to get from the pigs. But then this Eli Lilly company was able to commercialize this, was able to synthesize insulin in vitro. When I say in vitro, that means in the lab itself. We were not we were not taking it from any animal or any source. We were just making it. So how did they make it? So now we understand that in the type 1 diabetes people, there is no insulin. So what they did was, so basically inject insulin. We can give insulin. So that means insulin is a protein. We also know that. And if it's a protein, that means it must be synthesized by a gene, insulin gene, right? So this is our gene of interest. This is our gene of interest because insulin. Because for example, I am healthy. I do not have diabetes. That means my body is able to produce in a sufficient amount so that the glucose is not in, like, so that I'm not diabetic anymore. But the other person might have lost the cells that is producing it. So we can directly inject because at the end of the day, the role of insulin is to just go into the bloodstream and stimulate our adipocytes as well as skeletal muscle to take up the glucose in the blood. So basically, we can introduce insulin directly, right? So what they did was Eli company, they identified the gene of interest. They identified the gene of interest, right? And this was the insulin gene, insulin. And what they did was they basically cloned it in a bacterial vector, bacterial plasmid. They cloned a gene of interest. Now they wanted, so the ultimate goal was, what was the goal? Goal was to make insulin protein, right? So since in the bacteria, so since they have used the host was bacteria. They used a bacterial plasmid, and then after this, they basically transform, transform the host. It is the host. Bacteria is the host. Once this plasmid enters the bacterial cell, and this its its own genome, and this is the gene of interest containing the plasmid. So what once it enters, all the enzymes, tacpol, uh, all the like machinery of protein and like translation and transcription comes into the play, and they basically make a insulin protein, insulin. Right? This is the entire process. So based on my ultimate goal, what I'm planning to do, what is my end goal of with the gene of interest, I'll be shortlisting or even selecting the gene, selecting the host. So that is the thing. That's the uh, third tool of the recombinant biotechnology. So we identified the insulin gene, and then we used molecular scissors to cut both the bacterial plasmid as well as the insulin plasmid, the insulin gene, and then we pasted it. And after pasting, we chose that since it's in the bacteria, we chose bacterial plasmid, and then we transformed into this bacterial. See, as you can see, there is no plasmid here, but you can see a plasmid, a plasmid, right? So this is what the third tool is, because even after ligating, even having this, but if we don't have the if we don't have the host, then we can't do it, right? So that is the third important tool of recombinant biotechnology, recombinant RNA technology. That is the host. Now, I was talking about the host, but here they have used a special term that is competent. Competent, like if I say it in Hindi, it's like saksham. So there, if we're talking about the competency of the host, but in what respect? Like, I mean, is it like when we tell someone that someone is competent or not, we are like basically uh, are trying to like understand the attribute based on the whatever he is or she is. That's what the competency is. So what when I'm telling that this is the competent host and this is the incompetent host, let's say if I am saying that I'm talking about host, and this host can be competent, can be incompetent, competent, competent. But on what criteria and what basis am I telling whether it's a competent or not? So this competency is defined as it's the ability of the host to take up the plasmid, right? So I hope you understand this. So any host, like bacteria, as we already know, we know that bacteria is everywhere. So this all these bacteria take up DNA or anything that is there. Like for example, when we say some virus or some communicable disease are airborne, so that means it should have like it has some kind of bacteria, some kind of DNA, like a lot of molecules. So, but they don't take it because there is something called competency, and this is what we are going to study. We need to understand how this competency is and what this competency is. So we are talking about a competent host. It's the ability. What is competency? It's the ability of the host to take up the plasmid or any DNA basic test for that matter. So this is what competency is defined as. So this is what competency is defined as. So moving on. So this is the third tool of RNA technology. First is the we have already studied about molecular tools. We have studied about the molecular scissors. Then we have studied about the cloning vectors. And the third thing is the host. Because after ligation, after preparation of the gene of interest in the plasmid, we have to introduce this plasmid in a suitable vector based on our gene of interest, like based on what we desire. That example that I gave was about insulin. Insulin we want to produce at a larger scale, and bacteria is one of the suitable organism or host organism which can produce protein at very large amount at a very cheap rate. So that's why the Eli Lilly company was able to synthesize insulin in vitro in the lab, and they were able to pioneer and then patent this process. So now we were talking about the competent host. Competency is defined as what? Competency is basically ability of the bacteria to take up the DNA, right? So that was, as you can see, the definition is also here. So now, since DNA is a hydrophilic molecule, it cannot pass through cell membrane. So I hope you understand the word hydrophilic and hydrophobic. So basically, DNA is a hydrophilic molecule, it cannot pass through cell membrane. Now, what is this? Like, I mean, we came across a term hydrophilic, right? Hydrophilic. The second term is hydrophobic, right? So I think you might have learned about this thing. So it is like it's in the name itself. Like hydro means water, and this philic means loving, right? And again water, and this is hating. Now we have a bacterial cell wall, right? Now as I told you, like this is a normal bacteria, right? This is not a competent bacteria. This is just a bacteria. So this cell membrane is made up of. We know that the most common is bilipid layer, right? Bilipid layer. So this they do not have lipid layer, bilipid layer, but they are made up of lipids. This plasma membrane is made up of. This is in eukaryotes, and this has a lipid layer, and a bit composition is different. So this is the lipid, and we know that lipid is hydrophobic, and now we understand that DNA is hydrophilic. So this hydrophilic, they are not able to pass easily. This is what they are trying to say. So when I say one more thing, like let me introduce another term for you. That hydrophobic, like hydro means water, phobic. So whichever is hydrophobic, that means it will be lipophilic. Since it's water eating, that means it is a lipid loving. So it's a given, right? So if someone is mentioning that this compound is a hydrophilic compound, that means it can easily dissolve or interact with water. You should automatically accept that this compound will not be dissolved or like mixed in lipid. Lipophobic. So now, since DNA is hydrophilic and the membrane itself is hydrophobic, that means it cannot pass easily. So the bacteria cell must be made competent. See, this was about the normal bacteria. In a normal bacteria, there is no competence. There cannot take. They have inability to take up any DNA or any foreign material. The next thing is like, so the bacteria cells be made competent. They must be competent enough to take up alien DNA or the plasmid, right? So this is where the competency is, the ability of the host to take up the DNA or the plasmid. Now, how to make a competent cells? So the process is quite vigorous and then very important. So for this, the bacterial cells are treated with a specific concentration of divalent cation, calcium, calcium. So the DNA enters the bacteria through pores in the cell wall, right? See, we knew like, okay, now we have understood that normal bacteria are not competent. They are made competent after treating them with a specific concentration of divalent cation called calcium. And then the DNA enters the bacterium through pores in the cell wall. Now you should understand one thing that that this is a DNA, or let's say this is a plasmid, right? This is taken up by E. coli bacteria. So the special name is DH5 alpha. DH5 alpha is the strain of the bacteria which have been altered so in such a way that it won't affect the gene, whatever the gene given. For example, we know that mutations and recombination very common in our body, but this bacteria that is DH5 alpha, one of the strains of competent cells, is have been they are mutated. Well, like the enzymes that can mutated the enzymes, because how to, like, for example, we know that in our day-to-day life in our body, like there are a lot of mutations occurring on the daily basis, on the hourly basis, basically. So how do I know that the gene of interest, whatever I have given, is same even after in the bacteria? What if the bacteria mutated it? Because again, because it will translate, even they will, like, since we have studied about the copy number, they will amplify the plasmid in its own cell autonomous self-replicating already they are. So how do I know that whatever the gene I have given, it is right or not? Because any mutation in the gene, any kind of mutation, deletion mutation, frame shift mutation, point mutation, anything will lead to a different product. Therefore, what we do is we have the scientists have designed and understand and basically given one of the best competent tool, that is DH5 alpha, where they have mutated all the enzymes which was responsible for causing mutation on a such thing, right? So this is it. Now this process of putting the plasmid into the E. coli is called transformation. So that's the process of transformation. So what is the transformation? Transformation is the transfer of naked DNA into the host. So that's transformation. So now we were discussing of competency, right? So now we understand that this is a normal DH5 alpha. Now the plasma membrane of the bacteria is negatively charged. Then we have DNA. DNA is also negatively charged. So there is no property for them to basically for this DNA to go in. So what we do is we use a divalent cation, that is calcium. And then what they DNA to come in contact, since it's already 2 plus, so they will basically attract each other and they will come in close contact to each other. Then we'll basically, after they come in contact with each other, we'll give a 42-degree shock. This is called heat shock. And once we give this heat shock, there will be four openings on the cell wall itself, and the DNA will automatically go in. After that, I will keep it on ice, and they basically will close. Right. This entire process is called transformation. So first we have this normal cell, and it won't take up the DNA. So we'll make them first as a competent host. So so what we do is basically we'll first take up the DS5 alpha cells. We'll treat it with calcium for a while, and then that way the calcium will be in the media itself. And then when we add DNA, this DNA will come and stick on the walls of the bacteria. And then we'll give the bacteria at 42-degree. We'll give it a shock because it's all in ice. And then second, we give it at a 42-degree shock. The pores will open and the DNA will enter. And then in order to close the pores, we'll again keep it on ice, and therefore the pores are closed. And that's how this thing will go in, the DNA. So now we have understood that DNA is a hydrophilic molecule and cannot pass through cell membrane because cell membrane is hydrophobic. So the bacteria must be made competent, so for which we use basically calcium treatment. And after calcium treatment, since both of them are negative, the DNA and the cells are negative, we give the calcium, and then they can basically interact or attach to itself. Then we give them a 40-degree shock, and that opens the pores, and from there the DNA enters, and then we again close it. So that is a transformed bacteria. So this is the entire process. So this is the preparation. This is how you make the competent cells. The preparation of competences, and then these are normally stored at -80 or liquid nitrogen because they are very sensitive. After this preparation, we'll add the DNA. The DNA, it will go and stick to the walls. And the second we give the heat shock, and this DNA will enter inside. Moving on. So such cells are incubated with recombinant DNA on ice for 30 minutes. Everything is standardized. 30 minutes, the DNA, the primer and the DNA will come into contact with each other. Then they are placed briefly at 40-degree. So this is 30 seconds. 40-degree, and then put on ice. This for 5 minutes. 30 seconds. So first incubation ice first is normal cells. Then we treat it with calcium chloride, and therefore it gains competency. And after competency gaining, we put the DNA and incubated ice for 30 minutes. For that, the DNA sticks on the walls, and then we give them a brief heat shock 40-degree, and then the DNA basically 40-degree for 30 seconds, and then the DNA goes in because the pores are open. And then once we keep it on ice, the pore closes, and then we basically the bacteria is transformed. So this was the whole method of DNA transformation. Now another thing is other methods. That was the transformation. Other methods to introduce the DNA into the host. So there are so one was transformation is one method that was used to transfer the gene of interest into the cell. There are other methods as well. One is micro-injection, one is ballistic gun or gene gun, and one third is disarm. Sorry, I was disconnected. So what I was telling you is that there are various methods. There are other methods. So apart from transformation that is used to in transforming, sending the DNA into the bacteria, there are other types of transferring of DNA. First is microinjection, then we have gene gun, then we have disarmed pathogen vectors. So what happens here is in the microinjection, the needle size is very low, and even this animal cell, they are around 50 micro to 100 micron. This is very small cells. Like you won't be able to they're basically your cells. So now you can correlate that you cannot see the cells itself, and at that level we work and inject the DNA. So in this microinjection process, the recombinant DNA is directly injected into the nucleus of the animal cell. Then we have gene gun, where the cells are bombarded with a high velocity microparticles of gold or tungsten coated with DNA. This method is suitable for plants. So this is the gene gun. So it's very clear, like they are basically in this gene gun process, the gold or tungsten particles are coated with the DNA, and then on the plant cell that will be kept on the slide or somewhere, they are basically bomb blasted. And so basically, when they bombard or hit each other, the DNA is transferred into the cell. I know might look unrealistic, but people have tried such many things, and it has been still followed for the plants. Then third one is the disarmed pathogen vectors. That is when it infects the cell, the transferred the recombinant DNA into the host. So we have already studied about this. We know about agrobacterium fumification. So these are the three main things. Three other things. So let's have a recap on this one. The host. So when we talk about host, always remember we talk about DH5 alpha. Okay. Now to make this host, because now this host, that is this is also E. coli only. Now this host has to take up the DNA. So for that we need to make host competent, right? To make the host competent, we basically treat it with calcium, and when it is done, then it is stored in -80. Like it's a very you can understand that your fridge is 4 degree, and in the fridge there is a unit where ice basically uh, forms, that is -20. So I am talking about -80. So this is what this is how the comp cells are stored. So now the calcium is added, and this calcium, I have already told you, like basically the DNA and the both are negatively charged. So the calcium is the one that can bring them together. So once this calcium comes in, and then when you add okay. So when you have a competent cell, this is an append of like a tube basically. So this is the thing. This around 50 microliters you will have. So in this, what you'll have is basically a bacteria with calcium all over. Now this is a cell. Now when you add DNA, what happens is in the bacteria, the DNA comes and sits here. Once it sits here, we'll give them a 40-degree shock, and the pores will open, and then this DNA will go inside. 40 shock, the pores will open, and you will have the DNA inside the cell. And then we'll just let this bacteria grow, and then plate it. On the plate, you'll say multiple form. So this is the entire process of transformation. So after this, making them whole competent is basically the ability of the bacteria to take up the DNA, and then we give a 40-degree heat shock because these are on ice, literally on ice, and this is what it is. Now, this is one method. Then we learned about different other methods. Uh, okay, let's have one different topic on this methods of DNA transfer. So first, you have already learned about transformation. So bacteria. Then you learned about micro injection for bacteria. This is animal work, animal cells, the DNA into nucleus directly. Then we have ballistic or gene gun, DNA to plants. Then we have third thing that we learned was about unarmed vectors, and we all have also already learned about TI plasmid, which is causing a disease called crown gall in the plants, and this is what. So now moving on, we have completed one section of the entire chapter 9 chapter, chapter 9. We have completed one part of this chapter. Now I'll be giving a brief revision on what we have learned, like a basic summary of each topic, so that you can have a grasp on what you have till now covered. And after this, we'll be solving few questions, and then in the next class, I'll be teaching you further molecular tools of how to isolate DNA and what are the techniques to amplify and everything. Right, now, let's see. So we have understood what is biotechnology. So biotechnology is basically a techniques. Technology is the techniques of using live organisms or their enzymes for products and processes useful for humans. So biotechnology is defined as the technology or the techniques of using the live organism or the enzymes to make the product. Okay, technology which uses bio, that is live organism, in order to produce product, and this is useful to humans. So according to EFB, it is basically the integration of natural sciences and organisms, cells, parts thereof, and molecular analogs for product and services. So this the natural sciences is basically technology, organisms, everything is the biology part, and this is an amalgam. So they come together in order to produce so many items or uh, so many items for the service of the humans, right? Then we studied about the basic steps in genetically modifying organism. First is to identify our gene of interest. First is to identify our gene of interest. This identification of DNA to identification of DNA with desirable genes. Okay, right. So, for example, I already gave an example of gene of interest, insulin, and how it was made of gene of interest and how it was cloned and done. So first we identify what we want. That is, let's say we want insulin. Then we'll identify the host in which the insulin protein can be easily amplified and synthesized. So that is, then we'll identify the host. And third, in order to maintain this host, the gene of interest, third basic steps is to maintenance of the gene of interest. First is to identify the gene. Second is to introduce the gene in the suitable host. And third is to maintain that gene in the host, right? So these are the three basic steps, and we have already understood what is the difference between the traditional biotechnology and this gene therapy, this RDN technology. In the traditional biotechnology, we have already understand that uh, undesired products are produced, but in this, it is not the case. So this is it. Now, then for the last few classes, we were discussing about the tools of recombinant DNA technology. We learned thoroughly on molecular scissors. Then we learned about the resistance enzyme. Then we learned about the cloning vectors, and then we recently studied about the competent host. Not host, it's a competent host. Again, very important. So, so in this class, we completed all the three tools. Now, let's solve your questions and understand, so that you can understand what all you have learned and maybe gain confidence from that, right? So, first question, how is the number of plasmid vector and yield of the recombinant protein related to each? So they're trying to ask us how is number and yield of protein. What is the relation between the copy number and yield, right? Now, again, the central dogma of life. Central dogma. Let's say there's one DNA, one gene, and from the gene we'll get a mRNA. From mRNA we'll get a protein. From the mRNA we'll get the protein. Right. Now, how is copy number defined? Copy number is basically it is the ability of the plasmid to replicate. So how is so it's basically the ability of the plasmid to replicate, right? So now what we have understood right now is that we know that the copy number is all about the plasmid and not the gene of interest. So if you introduce the gene of interest, if you introduce the gene of interest in a plasmid where the copy number is very low, so we have already understood about the copy numbers. So what if, like, for example, you have two different vectors, like, let's say vector 1 and vector 2, right? Vector 1 and vector 2, and in this vector the copy number is, let's say, 10, and this has 100. Now you will obviously choose the vector that has a higher copy number. Why? Because you understand that for every gene there will be some amount of RNA made, and from this RNA, again, each RNA will give us a lot of protein. So this it is basically directly proportional. the more the copy number. So what is the thing? Like the higher copy number means higher will be the higher copy number of the plasmid means higher gene of interest. And if the goal is to make insulin, like the protein itself, since there are a lot of genes, that means it will express and it will give us a lot of protein. So that is the relation. So the relation here is basically basically the copy number of the plasmid is directly related to the yield. Copy number is directly related. uh, the conclusion from this is copy number is directly proportional to the yield of protein. Higher the copy number, the greater is the higher the copy number, the greater is the copy number of the gene, and consequently, the greater will be the protein. So, this is the concept of this. Second question: can exonuclease be used while producing a recombinant DNA molecule? So, we have what is exonuclease? They are basically again a molecular scissors. Here, molecular scissors. And what is that? We have learned about two types: endo and exo. We know that endo, both are restrictions and basically identify a particular sequence and they cut the DNA. So endo basically will cut in the middle, while the exo will cut, like this is a gene of interest, so it will cut at the end, right? So now the question is, can exonuclease be used while producing a recombinant DNA molecule? And the answer is no, because they won't cut properly first, like they the DNA, and they cannot they cannot produce the sticky end. So I hope you have understand that what is exonuclease. Exonuclease is the molecular scissor or a section enzyme that basically cuts DNA at the end, and they won't produce right. So first, it cannot be used in RNA technology because they can degrade the DNA because they cut at the neck and they cannot produce the sticky ends. Moving on, what are competent cells? What does the word competent mean? Right. So this is what we have recently, like we just learned about 20 minutes before. So what is competent cells? They are cells that allow the foreign DNA to incorporate to the host. Competent cells are basically cells that take up the DNA, or basically they basically allow the foreign DNA to incorporate into them. Right. Now, what does the word competent mean? Right. So I've already discussed about this. It is basically the ability of a cell to intake the DNA. So first thing where we learned is the competences. What are the competences? They are basically the DH5 alpha. Always remember that this is DH5 alpha because it is a type of E. coli. But the catch here is that this strain of E. coli has been removed of the enzymes that can deteriorate or affect your gene of interest. For example, we know, like as I already gave you, that any gene is prone to mutation, recombination, anything can happen. So that like what if you gave a functional gene into the DH5 alpha, and if the mutation gene is not inactivated in the DH5 alpha, what if it mutated the insulin receptor, insulin gene? So you won't get the desired product. So that is why we use DH5 alpha, which is a uh, which has the genes, sorry, the enzymes that are causing mutation. They're basically removed or mutated basically. Now, competency or competent refers to basically it's the ability of the cell to take up the DNA molecule. So that's the competence. Now, what are the features of the plasmid being used as a cloning vector? This is a very big question. Like, I mean, this is a very like 5 marks question or something like that. So you have to understand and you have to mention all the features of a plasmid that can be used as cloning vector. So I hope you remember the example of PBR322. PBR322. So what are the features of the cloning vector? First, it should have a ORI. What is ORI? ORI is basically origin of replication, because you want your plasmid to replicate much more often. So there's a gene of interest is replicating at a higher rate. And whatever, if you want a gene, if you want a protein, you will get in abundance. So this basically defines your copy number as well. So first is ORI. Second, we need is a selection marker. Selection marker. So we have already studied about antibiotic markers, antibiotic selection markers, where it will have the resistance, and if we grow that, for example, if the plasmid has amp resistance, that means it can degrade amp, so they can easily grow in amp plate. Then we also learned about blue-white screening. I hope you remember that, blood screening, so that we can remove the two-step process, right? So first is ORI, second is selection marker. Now next, the third thing, very important, it's the MCS, that is cloning sites. So we need restriction sites, restriction enzyme sites, right? Because of this, we'll be able to incorporate our gene of interest. So what and all we have? We have ORI, then we'll have MCS. MCS is basically a hub, like it's a DNA sequence wherein there will be lot of enzyme sequences there. So even in the even 100 base per repeat, we'll have around 5 to 7 enzymes as we can easily clone. Then we'll have a selection marker, and here in MCS side we can introduce our gene of interest. So these are the features of the plasmid, right, which can be used as a vector. Moving on, what do eco R and I refer to in the enzyme? So this is basically here they are asking about the nomenclature of the enzymes, nomenclature of restriction enzymes. So you already know this. So eco means refers to the species. This refers to the strain, and one means the order in which the enzyme was isolated or identified. So in this vector, in this strain, in this bacteria, this was the first enzyme. Eco R1 was the first enzyme to be done. So this is like E is the genus and coli is the species. So this is what it is. And then moving on to the next question. So okay, now next question is the biotechnology that you have already we have already defined it. So biotechnology is basically uh, it's basically a broad area of biology which uses both technology uses both technology plus technology and organism to develop and products produce products to for human welfare. So this is a normal thing that you should remember or understand basically, because they don't ask this kind of questions much. So you should just understand, if someone tells you about biotechnology, you should understand that it is basically a broad area of biology, and this is part of biology where the technology where the technology and the living and the organisms are used to develop and produce products which will be useful for the human welfare, right? And yeah, and this word was coined by what is that guy, something like Caroli okay in 1990. Moving on, what is genetic engineering? Again, it is a technique. Genetic engineering is a technique used to change or modify genetic material, such as material such as RNA or DNA, to introduce them into the host. So genetic engineering basically is a manipulation of the genetic material itself. It's engineering or the manipulation of genetic engineering. Sorry, of the gene itself. So moving on. So basically, genetic engineering is a technique used to change or modify the genetic material in order to introduce them into the host and get our desired product. Now another question is, how is TI plasmid of Agrobacterium tumefaciens modified to convert into cloning vector? I hope you remember the TI plasmid. So again, this Agrobacterium tumefaciens causes what? They cause one disease called crown gall disease. It's a type of tumor in plants. Basically, a tumor, a cancer in plants, right? So how they do it? They have this TI plasmid, and in this TI plasmid there is this tDNA, or the transfer DNA, which goes tDNA gets incorporated to host, right? This is what the tDNA. So what scientists? So the question is, how is the TI plasmid of Agrobacterium tumefaciens modified to convert into a cloning vector? So you just have to answer that the tDNA, which is causing the disease, is removed, and the gene of interest is incorporated in that place, incorporated in that place, and that's how it is used. Moving on, how are competent cells were prepared by action of like calcium chloride. So this calcium chloride first makes DNA in contact with host, and second helps in opening of pores in host so that so that DNA can easily enter in. Right. So what is the importance of calcium chloride? Calcium chloride first it gives the polarity to the DNA as well as the components so that they can come in contact with themselves, and then they're upon heat shock they are also helpful in creating pores in order in the host so that the DNA can easily enter. So what are the principles of biotechnology? We already know this. This is a straightforward answer. It's a genetic engineering and bioprocess engineering. So genetic engineering deals with manipulation of DNA, while bioprocessing basically provides environment provides safe environment like autoclave, like safe or sterile environment to work in. Now another question similar to the previous one: what is the role of agrobacterium purification in plant transformation? So we know that agrobacterium purification is what it's basically a plant pathogen and a soil-borne bacteria, right? And it infects many plants, it infects crops such as tomato, cotton, soya bean, etc. And it causes what it causes? We already know it causes crown gall disease, which is because of TI plasma in the spirit tDNA. So this is what they wanted to know. What is the role of agrobacterium in plant transformation? So this is how they do it. Now, how is biotechnology useful in developing food crops and agricultural processes? Now, this is basically a general question, but I hope you must remember the thing BT cotton, a cotton plant, where the genetic by using genetic engineering, the toxin gene from BT, that is Bacillus thuringiensis, was incorporated into the cotton genome, and therefore we were getting a BT cotton. And that's how the biotechnology is very important and has been playing a very important role in developing food crops and agricultural processes. Even though India is not accepting such crops much, like I mean, not accepting it profoundly, but like the crops is like because apart from BT cotton, there was another crop that was made that was BT brinjal, and due to other since it's a GMP plant that is genetically modified plant, people are having like they do not intend to consume it, and therefore the BT brinjal was basically stopped. And since cotton is not a consumption product, people are accepting that. So even though biotechnology has the immense potential to help us uh, in our food as well as agriculture sector, the mindset of our like our generation basically or the farmer generation is not that apt not to accept that. So that's why we are not using the recombinant biotechnology, but at the base level we have been using biotechnology from the day one of this like day one of the agricultural products, because anything like if you search for example maize, any food item that maize is one of the most common example in the before biotechnology came in, before the crossing or before the hybridization took place, they were like very thin, very different colors, and then they were like I mean like not consistent. But now, as you see, that they are like a corn is kind of big and it is very sweet and yellow. It is all because of the hybridization, which is also a part of biotechnology. So people are accepting biotechnology, but they are not accepting the way it is done. So in the normal one, we just take seed from a better plant and then sow it or like pollinate it, and we'll get a better product. So that's how the biotechnology is playing a very important role. So next question is, what do you understand by gene cloning? Right. So again, this is a straightforward question. So in gene cloning, what is like gene cloning? It is basically a process wherein the gene of interest is uh, isolated from a DNA and then is cloned into a plasmid and then given into a desired host. Desired host. So what the entire process basically gene of interest isolation, the isolation of DNA fragment, then putting it into a vector and then into a host. So I have few more questions that we'll discuss tomorrow, and then we'll be then then we'll be starting another topic. So thank you for joining today. Uh, in the next session, we'll continue with again the same question answers part. I'm having around more 10 or 12 questions, and after that, we'll be learning about how to isolate DNA and how to basically like amplify that DNA and that kind of process. So thank you for joining. Let's meet in the next session.